All right, let's jump right into a huge biological puzzle. How in the world do scientists find one specific gene when it's buried inside billions of letters of DNA? Well, the answer is this incredible tool called a DNA library, and we're about to break down exactly how it all works.
So let's start with the big problem, right? The human genome. It's just this massive, massive string of information. We're talking over 3 billion letters long. So how on earth are you supposed to pinpoint one tiny little segment, a single gene, that might be the key to a certain disease or a specific trait?
To really get a feel for this challenge, think about it like this. Imagine you have the greatest recipe book ever written, but it's been chucked through a paper shredder. All the words, all the ingredients, the instructions, they're all just mixed up in this one giant, chaotic pile. That's pretty much what you're up against with raw DNA. And here's the size of that pile, over 3 billion base pairs. It's just a staggering amount of data. I mean, to put that into perspective, if you tried to read it all out loud, it would take you almost 100 years. So yeah, without some kind of system, finding anything specific is, well, it's basically impossible.
So, what's the solution? Scientists came up with something brilliant, the DNA library. The idea is pure genius. You take all those shredded pieces of DNA and you clone each little fragment into what's called a vector. You can think of a vector as a biological delivery truck, like a tiny virus or a plasmid that can carry the DNA. This creates a whole collection where each book in your library is now a single, manageable piece that you can copy and study. It's the ultimate filing system for our genes.
Now, there are two main kinds of these libraries. We're going to start with the big one, the most complete version, the Genomic library. This is like having the full, unabridged blueprint for an entire organism. And that leads us to our first big question. What if your goal is to have a copy of every single piece of an organism's DNA? I mean, the whole shebang, the complete picture. If that's what you're after, then a genomic library is exactly what you need. And when I say everything, I mean everything. It has the genes themselves. Those are the protein coding parts we call exons. But it also has all the non-coding sections, the introns, which are kind of like the filler material between the actual instructions. And, crucially, it also has the regulatory sequences and promoters. Those are the bits of DNA that tell genes when to turn on or off. So how do you actually build one of these libraries? Well, it's a pretty cool process. First, you pull out on the DNA from an organism's cells. Then you use these special enzymes to chop it all up into random overlapping pieces. Next, you insert these fragments into a vector, often a type of virus called a bacteriophage, which is really good at carrying big chunks of DNA. And finally, you let these vectors infect bacteria, which then act like tiny little copy machines, making millions of copies of each DNA fragment. And what you're seeing here perfectly illustrates that process. You can see the human DNA getting chopped up, slotted into the bacteriophage's own DNA, and then packaged up into new virus particles. Each one of those viruses is now carrying a different piece of the human genome puzzle, creating this huge, searchable library.
Okay, let's switch gears and look at the second type of library. This one builds a very different kind of collection. It's called the cDNA library, and it's not the complete blueprint. Instead, think of it as a snapshot of what a cell is actually doing at a very specific moment in time.
And this brings us to our second key question. A cell in your liver and a cell in your brain have the exact same genomic DNA, right? But they act totally differently. So why is that? It's because they're using different sets of genes. So, what if you only care about the genes that are switched on right now? To do that, you can't start with DNA. You have to start with something called messenger RNA, or mRNA. These are basically the temporary, disposable copies of genes that a cell makes when it needs to build a specific protein to do a specific job. So if you look at all the mRNA in a cell, you get a perfect picture of exactly which genes are active. But there's a little problem. RNA is pretty fragile, so scientists use this incredible enzyme called reverse transcriptase. And it does something amazing. It literally works backward. It uses the RNA message as a template to build a stable, double-stranded DNA copy. And we call that copy complementary DNA, or cDNA. Now, what's so important about this is that it shows a crucial difference. See, in our cells, that initial RNA gets processed. All the non-coding bits, the introns, get cut out, and the important coding parts, the exons, get stitched together. This means the final cDNA you make from it is a super compact, protein-coding-only version of that gene, no filler. And the process to build it reflects that. First, you isolate only the mRNA from a specific tissue, say a liver cell. Then you use reverse transcriptase to make that first DNA strand.
After you get rid of the RNA template, another enzyme comes in and builds the second DNA strand to match. From that point on, it's the same old story. You pop these cDNA fragments into vectors and create your library.
Okay, so we've got these two really powerful but very different tools. Let's put them side by side and do a direct comparison to really nail this down. This table just makes everything crystal clear. A genomic library, it starts with all the DNA. A cDNA library, it only starts with the active mRNA. That means only the genomic library is going to have those non-coding introns and those super important regulatory promoters. So here's the bottom line. A genomic library represents the entire potential of an organism. It's the whole giant cookbook. A cDNA library, on the other hand, represents what that organism is actually doing at one moment in one place. It's just the recipes you're cooking for dinner.
So we've seen how these libraries get built. But why does any of this really matter? Well, the applications are where this technology becomes truly revolutionary, changing everything from how we do basic research to how we practice modern medicine.
The uses are just incredibly broad. With genomic libraries, we can study the very structure of our genes and how they're controlled, which is fundamental for understanding genetic diseases. With cDNA libraries, we can do amazing things, like compare the active genes in a cancer cell versus a healthy cell, helping us figure out exactly what's gone wrong. We can find brand new genes, trace evolution across species, and this is a big one. We can use these libraries to turn bacteria into tiny factories that produce life-saving medicines like insulin.
And look, it's really not an overstatement to say this stuff is a cornerstone of modern biology. It's the foundation that so much of our genetic knowledge is built on, and it's still an absolutely essential tool for discovery today.
So by creating these organized, searchable libraries, we've basically taken that shredded-up book of life and turned it back into something we can actually read and understand. And that leaves us with a pretty exciting question. Now that we have the library, what new chapters of biology and medicine are we going to explore next? Hey, if you found this breakdown useful, make sure to hit that like button and subscribe for more deep dives into the world of science. Thanks for watching.